Okay, good morning, dear guys. Let's continue our set of lectures about immunology. And in last topic, we stopped in cell-mediated immunity, activation of T lymphocytes, and elimination of intracellular microbes. And I want to remind you one main point. We have two main types of T lymphocytes. It's CD4 positive and CD8 positive. So CD4 positive helper T cells, so they're CD4 positive, they are usually helper T cells, and they are responsible for recognition of antigens from the MHC1. Oh, sorry, MHC2, a main histocompatibility complex type 2, which is presented by antigen-presenting cells. As we discussed before, they are like their dendritic cells, yeah, so and Langerhans cells, they're typical antigen-presenting cells, but also MHC2 is present in B lymphocytes, and we will discuss it today a little later. But in the CD4 positive helper T cells, they're not responsible for killing cells. They're responsible for to produce cytokines like interleukin 2 and also expression of and they have also receptors for interleukin 2. So you see, interleukin 2 causes it's a like growth factor, so which acts on T lymphocytes and stimulates the proliferation of the cells. So you see, they're like self-stimulating cells. They stimulate itself and also other T lymphocytes. Okay, functions of helper T cells are mediated by the combined actions of CD40 ligand and cytokines. So CD40 is a member of the TNF receptor family. So you remember we have TNF, and CD40 is acting after the TNF activation. So TNF is produced by macrophages as a response to injury, as a response to bacterial invasion, as a response of inflammation. Yeah, and that's any inflammation promotes the secretion of TNF and promotes the activation of T cells. Okay, when CD4 positive helper T cells recognize antigens being displayed by macrophages or B lymphocyte, I said you, antigen presenting cells, T cells express CD40 ligand, which engage CD40 on the macrophages or B cells and activates the cells. So you see, it's like the CD4 positive cells is like mediator, so which it's like it's confirming the process of activation of lymphocytes. Okay, let's continue in 1 minute. Алло, да? Слушаю. Смотрите, да-да-да, я понял. Смотрите, я уже с Айбеком, да, вы эту войну ведете, я так понял? Или нет? Не с Айбеком войну ведете? Потому что Айбек, он этот, смотрите, такая ерунда. Надо здесь договариваться с другими вузами, потому что я сам точно не знаю, как это делается. Я примерно могу сказать, но видите, если сейчас я примерно скажу, мы примерно сделаем, а потом Санпин придет и нам скажет, закрывайте вот это все, закрывайте всю байду, нельзя работать. Нам надо связываться с КГМА или КРСУ, где у них есть виварий, официальный, уже подтвержденный виварий, без каких-либо проблем. Нам оттуда желательно какого-нибудь специалиста взять, если у них кто-нибудь есть на примете, и чтобы он нам разрабатывал, потому что я, честно, не особо знаю, какие там приказы у нас по этому работают. Да, но у меня пара до 4:30. Если я до 5 успею. У меня прямо сейчас лекция идет просто, поэтому. Мгм.
Yes, sir.
So this mucosal epithelial cells, they secreted, so it also helps to eliminate the offending agent, especially if we talk about parasites or any other protozoas. And we will discuss it in the last topic in the semester. Okay, but let's talk about activated CD8 positive T lymphocytes. CD8 positive T lymphocytes, as you remember, might be forgot. They're known as CTLs or cytotoxic T lymphocytes. cytotoxic T lymphocytes. And as name implies, they're responsible for directly killing of bacteria. So CD8 positive T lymphocytes, they are activated by MHC1, first of MHC1 bound peptides. So we also discussed it in MHC, but I just repeat it again. And they are responsible to produce C cell receptors against these peptides which are bound to MHC1. And and then and they inside this cytotoxic T lymphocytes we have perforin and granzymes. They are stored in granules inside the cytotoxic T lymphocytes, inside the CT8 positive T lymphocytes. So and they and this you see perforin and granzymes, their function is this. So, okay, in easy words, they have attachment. They have on surface they have a T cell receptor, which is specific for some antigen. And if it finds some antigen, it attaches to this and releases perforin and granzymes. And perforin and granzymes, their main function is destruction of cell membrane, and they'll destroy the cell membrane of this bacteria, virus, so not virus, of course, bacteria, protozoa, or even the parasite. So it tries to kill it. So that's the main function of T lymphocyte. Also, they can kill our own cells with this with expression of this antigen. In cases of virus, for example, when a virus infect our cells, you know that viruses start to produce their own proteins, and these own proteins. And these proteins they are released by released with first MHC, and then this T lymphocytes, when detect this antigen, they kill the cell which presents this antigen on the surface, and that's the purpose of cytotoxic T lymphocytes. Okay, and also they can possess the receptors which activate the apoptosis process in the cells. In chapter number 2, we discussed the apoptosis, which it induces the apoptosis of target cell with the help of cytotoxic T lymphocytes, first of all. Okay. And one more idea that T cells are regulated by a balance between co-stimulatory and inhibitory receptors. We have co-stimulatory and inhibitory receptors. So major co-stimulatory receptor is CD28, and we discussed it. I said about it a little later, but of course you forgot about it. But CD28 is a main co-stimulator, and without activation of CD28, so cytotoxic effect doesn't happen. And especially, so for example. So protein CD, so this don't even remember. So for example, some tumors, they can produce anti-CD28. So they produce the specific protein which prevents the CD28 attachment to the surface, and these tumor cells can survive, because normally they secrete this normal kill me signal, so produced by apoptotic cells, by tumor cells, and these CD28 cells, CD8 positive cells, they can destroy these tumor cells with the attachment with CD28. But without this attachment, they survive, and that might cause the malignancy. So that's some malignant tumors, they avoid this attachment with CD28, and they survived. And later in next chapter, we'll discuss it, when we'll discuss the malignancy and so all the types of tumor, so all the neoplasia we have. Okay, let's talk about humoral immunity, activation of B lymphocytes, and elimination of extracellular microbes. So upon activation, B lymphocytes proliferate and then differentiate into plasma cells that secrete different classes of antibodies with distinct functions. There are two major pathways of B cell activation. So you remember B lymphocytes, are responsible for synthesis of immunoglobulins. So let's talk about these two. So here it's shown actually. Class switching, affinity maturation. So let's discuss it. We have two types. They're T cell independent and T cell dependent. So you see, B lymphocytes are pretty selfish, and they can pretty independent, yeah, independent from T cells, and they can be activized not only for proteins, not only with the proteins they can be activized. They can be activized just by polysaccharides and lipids and lipid antigens which are present in bacteria. So and they can independently activate the B cell activation, and specifically in this B cell activation, it's T cell independent cell activation. So it secretes the IgM, and that's why might be heard somewhere, but might be it's the first time you hear about it. IgM is the first response to against bacteria and viruses in our body. So IgM is the most rapid form of IgM, but most rapid form of antibody, and it facilitates the immunity reaction like first so like one of the first line of secondary immunity effect. But we have T cell independent T cell dependent reaction. So T cell dependent reaction is related to the function of CD4 positive T cells. As I remind you once again, CD4 positive T cells, they are known as T helper cells, and they produce cytokines to activize the B lymphocytes. So B cells APC, antigen presenting cells, because they possess MHC type 2 class, MHC second class molecules, and they just submitted to helper T cells. And then helper T cells express CD40 ligand and secrete cytokines which work together to activate the B cells. So you see, cycle is closed. Yeah, T cell dependent. So it's T helpers. So when antigen presenting, so when B lymphocyte presents the antigen, T cell confirms this antigen and then activates B lymphocyte to kill this antigen. Okay. And let's talk about types of antibodies which are produced. They are IgG, IgA, IgE, IgM, and IgD. Function of IgD is still unknown. That's why we'll discuss only four classes: IgG, IgA, IgE, and IgM. So IgM antibody is produced due to response to polysaccharides. You remember polysaccharides, so it's this T-independent pathway of activation. It causes the production of IgM. It's a first response against the bacteria or viruses for any invader. But later, when helper T cell is attached to this MHC2, then it can stimulate the B lymphocytes to produce IgG, IgA, and IgE. But it requires time. Activation by T lymphocytes requires time, and that's why this process is expressed a little later, so it's postponing, and that's fate of vaccination. That's why we need vaccination. So if our lymphocytes are naive to this bacteria, so to this infection, so we have to create our own new immunity, which requires time. But if we already had this immunity against it, so we can handle it in a fast way, so just in a few minutes. Okay, about the immunity after the vaccination, we'll discuss a little later. So main idea of so here this is not very important, but for instance, yeah. So this B cell, so which is activated by microbe, or might be activated by helper T cells or other stimuli. So it causes the proliferation of this B lymphocyte, and this proliferation of this B lymphocyte causes differentiation. And we have different B lymphocytes produce different class of immunoglobulins. There might be antibody secretion, function. First, antibody secretion due to microbe activation causes the formation of IgM. So that's a plasma cell which produces the IgM. So, but sometimes it causes a class switching, and this in this same cell which we used to produce IgM, so is targeted to produce IgG, which is more. So you see, IgG is more attachable, so more possessible, higher affinity to antigens, and that's IgG producing plasma cell produced. And then affinity maturation. So this high affinity Ig expressing B cells, after the maturation period, they also produce the high affinity IgG, and then they can kill this viruses or bacteria, doesn't matter. And this affinity maturation is this cell which happened due to affinity maturation, so and finally matured B lymphocyte. So it produces a memory B cell. Memory B cell it is a main goal of the vaccination. So this memory, so you see, immunoglobulin might be depressed, might be decreased in amounts, and then we like are not protected against the virus. So let's talk about coronavirus. Yeah, so it's the most abundant example right now. So in memory B cells, even if you ever if you had vaccination or if you had a disease as this coronavirus disease before, so you already have memory B cells. And gradually, so amount of these memory B cells are decreased. The memory of high affinity immunoglobulin expressing B cells is decreased, but memory B cells are remained for years, sometimes tens of years. So sometimes entire life they are present in your body, and your immunoglobulin level will be decreased. But when you encounter with this infection again, when one more person go without mask and then cough on you, yeah, so your immunity will activate this memory B cells. On the surface of these B cells we have immunoglobulin, yeah, and it is activated. It causes the activation of T helper cells, and T helper cells activate it again, and it causes the proliferation. So helper T cells activate the proliferation of this memory B cells, and they start to produce directly high affinity immunoglobulin. Without so this what is required. Okay. And then we have what's the functions of antibodies. So first of all, is this neutralization of microbes and toxins. This is known as this is yeah, just neutralization by attachment to the surface. Then of course next step is opsonization and phagocytosis. So this antibodies, they are used as markers for phagocyte cells or macrophages, and they can detect these macrophages and swallow it with the help of receptors. And we discussed it in opsonization topic process of um, in the process of complement system. So of course, complement system is also activized with antibodies. We'll discuss it here. And then antibody-dependent cytotoxicity, antibody-independent cytotoxicity. So we on the surface of NK cells we have FC receptor, so which directly can kill the offending cell. So with the help of with antibody attached to this FC receptor, so they use it like. So like a weapon, yeah. So just a weapon, so which you hold with the NK cell, and they directly killing this antibody-dependent cytotoxicity. So it's a typical feature of NK cell. Then, of course, complement activation directly causes lysis of microbes. You remember the C3b activation causes the formation of pores inside the bacteria. But if it's this same complement activation causes the phagocytosis of opsonized microbes. So that's the same idea as we discussed here. Yes, so in this. And of course, this antibodies, activation of antibodies with the bacteria, so joining of them, so causing the inflammation. Okay, and now so this main idea we discussed. Okay, and then of course we need to discuss the decline of immune responses and immunologic memory. So this long memory, so gradually after the production of a lot of antibodies, so we don't need to produce antibodies, and this most of lymphocytes, B lymphocytes, which are activized, they undergo the apoptosis process, and that's why in the end they remained only few cells which are known as memory cells. Yeah, we discussed it. So this summary you can read by yourself because it's summary of 2 lectures. And now let's continue to talk about hypersensitivity, immunologically mediated tissue injury. So what time? Oh, only 18 minutes passed, so that's why we continue. In immune responses that normally are protective also are capable of causing tissue injury, and this is known as hypersensitivity reactions or hypersensitivity diseases. So that's you already heard about this autoimmune diseases okay, let's talk about causes of hypersensitivity reaction. First of all, it's autoimmunity reactions against self antigens. For example, in our so you remember we have HLA so human leukocyte antigen, which produces self-tolerance. And when self-tolerance, self-tolerance process is injured somehow, we'll discuss it a little later. So it creates might create antibodies against it, might create C cell receptors on the surface of T lymphocytes. T cell receptors (TCR) might be produced on the T lymphocytes, and or NK cells might recognize it like antigenic structures, and they start to kill our own cells, which used to be normally toleranced. So, and that's we will discuss it. So, very interesting disease. Then reactions against microbes. So, for example, many types of reactions against microbial antigens that may cause disease. For example, examples. So just keep in mind that antimicrobial activity also can kill our own cells, and we will discuss it in this chapter a little later. And also reactions against environmental antigens. Environmental antigens, for example, this is typical atopy. Atopy, so it's allergy. So what we name as allergy. So this typical allergic reaction. So we have a genetic predisposition to this unusual immune responses. Now, so usually it's a response to harmless stimuli. Okay, we'll discuss it. So this no need to read it in details. Let's talk about classification of hypersensitivity reactions. This is main classification you need to remember. So that's so now this you must remember, please. So don't say that I give you too much topic, too large syllabus. So but everything else is nothing regarding to this classification. That's why this classification need to be understood totally. Okay, by types, we have immediate type 1 hypersensitivity, antibody-mediated type 2 hypersensitivity, immune complex-mediated type 3 hypersensitivity, and cell-mediated type 4 hypersensitivity. So you see, immediate hypersensitivity, let's talk about the immediate hypersensitivity. So immediate type 1 hypersensitivity called as allergy. So it's all of this is related to IgE antibodies, which present there. So how it's called? So it's a topic of continuous of discussion. Then antibody-mediated disorders. Antibody-mediated disorders, it's a type 2 hypersensitivity. They are caused by secreted IgG and IgM antibodies. So when antibodies are produced, and these antibodies are attached to our own cells and cause our own cell injury, our own tissue injury. So these antibody-mediated disorders. So large variety of the disease we have, and in therapeutic diseases you will discuss it again and again. So we have different types, but here we'll discuss the general pathogenesis of this disease. And then next one, it's immune complex-mediated disorders. Immune complex-mediated disorders. So as you remember, immune complexes they activize the classical pathway of complement activation. But what if these antigen antibody complexes they are present inside the blood? And what if they stuck in some organs? Especially they're usually stuck in kidneys. And these complexes might be stuck in kidneys, and they produce the clots. And in small capillary vascular beds, in capillary beds, they can cause the activation of leukocytes in this area, and also create the inflammation in this area, and cause the generation of toxic free radicals. So you see, it's inflammation without cause. So might be we have a cause, but its reaction is it's overreaction against it. And then last type, which is a most slow type of hypersensitivity, but it doesn't mean that it's less important. It's a type 4 hypersensitivity disorders. It's caused by immune responses in which T lymphocytes of the Th1 and Th17 subsets produce cytokines that induce inflammation and activate neutrophils and macrophages, which are responsible for tissue injury. And CD8 for positive CTLs, so cytotoxic T lymphocytes, also may contribute to injury by directly killing host cells. So you see, it might be T helper cell activized and promote the leukocyte aggression to to our own tissue, or might be direct activation of T killer cells to injure directly host cells. Let's talk about type 1 hypersensitivity. So immediate hypersensitivity is a tissue reaction that occurs rapidly, typically within minutes after the interaction of antigen with IgE antibody bound to the surface of mast cells. Take a look. If I'm not mistaken, it is here. So, for example, we have some allergen. So, for example, pollen. Yeah. So it might be it's so for everything allergy we have allergy for everything. So, for example, due to attachment of allergen to the surface of mucosa, so it gets swallowed by dendritic cells. And due to worst way of activation, so these dendritic cells present this antigen to the surface of naive T cell. You remember, it's in lymphatic tissue, it happens in lymph nodes usually. And this naive T cell is converted into Th2 cell, so a T helper cell and CD4 positive cell, and it expresses this antigen to the B lymphocyte, and it activates the B lymphocyte to produce this antibody against this antigen, against the allergen. And which type of antibody is produced? Especially for allergic reaction, it is IgE. Immunoglobulin E is produced. And this immunoglobulin E possesses specific feature. Immunoglobulin E is attached to mast cells. So on the surface of mast cells, we have special parts of receptors. It's FC binding ligands. So they bind the IgE like a ligand to the surface, and and then activation of this IgE causes the degranulation of mast cell. And as you remember that degranulation. So mast cells possess granules with histamine and heparan sulfate. Yeah, histamine and heparan sulfate. And reaction of histamine and heparan sulfate you already know: vasodilation, retraction and contraction of endothelium cells, and also and of course the increasing of permeability of blood vessels and causing all these effects. And it causes inflammation in some particular site if we have antigen in some particular site, and it causes a rash, for example, or might be rhinitis. Yeah, so nose. So for example, if your mast cells are attached with this IgE on the surface, so in nose only, so then if you smell something like pollen, yeah, it causes a degranulation of this histamine in this area. It causes an inflammation like reaction in the nose. Yeah, or might be in a skin, might be in a conjunctiva of eye, might be I don't know, so which other manifestations, so of allergy might be in the digestive tract. Yeah, so for example, immunity of digestive tract. But also if but sometimes in severe cases of allergic reaction, so when most of mast cells are attached with this type of IgE against some specific type of allergen, so it causes anaphylactic reaction, anaphylactic shock. This is a point we need to discuss. So it is explained here or not? So you see how much I skipped here. So that's because this explanation is enough to understand. So this and also, yeah, you remember that IgE binding with antigen, so causes the activation of eosinophils. Also eosinophils. That's why in cases of allergic reaction we have collection of eosinophils inside the tissues. That's part of morphology. So it's and this is. Okay, we have clinical and pathologic manifestation. It's immediate response, which is stimulated by mast cell granule contents and lipid mediators, and is characterized by vasodilation, vascular leakage, and smooth muscle spasm, usually evident within 5 to 30 minutes after exposure to an allergen and subsiding by 60 minutes. And second late phase reaction stimulated mainly by cytokines, which usually sets in 2 to 8 hours later, may last for several days, and is characterized by inflammation as well as tissue destruction such as mucosal epithelial cell damage. So main idea you understand. So first of all, we have immediate response, so by releasing of histamine. But second, late phase response, so it's when histamine already done their function, and also cytokines are released, and they promote the reaction, which is very similar to inflammation. Okay, This all this LTC, TH2, so don't need to remember. So just remember that eosinophils are activized here. So we have different manifestations. So let it be your individual, self-individual work. So this clinical syndromes which caused by the allergy, so anaphylaxis, bronchial asthma, allergic rhinitis, and food allergies. Let's continue. Let's talk about second type of disease. So type 1 disease is very easy, and in practical class I will ask you, but if you want able to answer, so we'll continue discussion of this. Then and don't forget to talk about anaphylactic shock. We already know what is shock, and in most of classes we already discuss what is shock, and then we'll discuss what is anaphylactic shock. So then antibody-mediated diseases. Antibody-mediated diseases, as you remember, so they're produced antibodies which are uh, directed against target antigens on the surface of cells or other tissue components. So against our own cells. So mechanism of antibody-mediated diseases is well known. So you already know what happens when antibody is attached to antigen. First of all, causing the opsonization and phagocytosis. So you see, due to opsonization, cell activity is interrupted, and phagocytosis causing the destruction of the cell. And then imagine our own cells, they destroying the cell. Also, don't forget about the complement activation. So due to opsonization, C3 splits into C3A and C3B, and C3B causing the attachment to the cell membrane, and it is like a signal to phagocytosis activation, and this opsonization happens, and that's why our own cells, our own components of our body, they get destroyed with help of phagocytosis. Okay, let's so we have these diseases. So also it's yourself individual work. It's autoimmune hemolytic anemia, autoimmune thrombocytopenic purpura, pemphigus vulgaris, vasculitis caused by ANCA, antinuclear centromeric antibodies, then good pasture syndrome, acute rheumatic fever, myasthenia gravis, Graves' disease, and pernicious anemia. So all of these autoimmune diseases, I guess, yeah, we'll discuss in practical class. So please, it is yourself individual work. Absorb it. So it's very interesting diseases. And okay, here we have some effects. So you see, opsonization causes phagocyte activation, and our own cell is get phagocytosed. Then complement activation causes the formation of pores inside the cells and causing the activation of leukocytes, like neutrophils, and neutrophils causing the oxidative stress in this area. And also, these antibodies can act on, if especially if we have antibodies against receptors, they can cause the impairment of receptor activation. And so here in example of C, so in myasthenia gravis, so we have antibodies against ACH receptors, acetylcholine. You remember that acetylcholine is it is a receptor for acetylcholine which is produced in nerves. So a nerve acetylcholine, which is produced by nerves, so it cannot activize these receptors because they're already filled with these antigens, and that causes weakness in muscles. This myasthenia gravis mechanism. Then and one very specific feature, so we have an antibody against TSH receptor. So and we used to think, yeah, if we have antibody against something, we destroy it. But this antibody works differently. This antibody starts to activize the TSH receptor. And this activation of TSH receptor, so you remember what is TSH, yeah? Thyroid stimulation hormone. And this thyroid stimulation hormone causes the overproduction of thyroid hormone. Thyroxine and triiodthyronine is increased because normally TSH is produced by pituitary gland. Yeah, and it stimulates the thyroid gland synthesis of thyroxine and triiodthyronine. But now we have additionally produced antibodies, and these antibodies they directly activize the thyroid gland cells. You already know what is thyroid gland, so from physiology, I guess. So that's why it's not a new thing for you. Okay, I guess type 3 and type 4 hypersensitivity is remained for the next class. Type 3 hypersensitivity we'll start from the type 3 hypersensitivity. So in next week practical classes we'll start to talk about immunology. So you already have this first lecture, so all the material of first lecture will be discussed there. So get ready, and if you want to get marks. Okay. See you next time. So let's check your attendance. Wait a bit, I'll check your attendance. Only 52 students joined. Okay, and according to the chat, we have much more students, of course. Okay, thank you. See you next time. You can leave.
Excuse me, sir. Excuse me, sir.
Yeah, Heather, any questions?
Sir, today you will take our practice class.
Today, yes, yes. So,
Sir.
okay, if there are
Okay.
no more questions, yeah, you can leave and we'll continue class. We'll continue the lecture set of lectures in next week.
Okay, sir.
Okay, sir.
Goodbye, sir.
Goodbye.